now you have to apply these genotypes and think about what they'll look like. Because remember, I'm talking about my phenotype here, my physical characteristics. So with my phenotypic ratio, I'm just going to count the number that look dominant. So in this instance, okay, so in this case here, I've got 1, 2, 3 that are going to look dominant. Okay, so I've got 3 that look dominant. And then I'm just going to count how many, my number that look recessive. And remember, to look recessive, they have to have they have to have 2 recessive alleles to actually look recessive. So this is my only one that's going to look recessive. So I've got a 3-to-1 phenotypic ratio. So again, all I'm doing is I'm just counting how many of them look dominant versus how many look recessive. It is extremely important that you slow down when you're doing this stuff and pay very close attention to whether it wants genotype in the actual alleles or whether it wants phenotype. Same with on the last, just a minute ago, when we talked about percentages versus fractions versus ratios. You have to slow down and know what it's asking for. Okay, so let's look at a couple of sample problems here. Okay, so what I have here that is in pea plants, smooth peas are dominant to wrinkled peas. If 2 plants that are heterozygous for pea shape are crossed, what would be the ratio of genotypes in the offspring? So my 2 plants that I've been given are heterozygous. It says both of them are heterozygous. So that means both of them have 1 big, 1 little, and that's crossed with another one that is 1 big, 1 little. So that's my first step. Okay, step 1 is I need to set up my cross. I need to know what my mom and my dad are. What am I doing? Okay, so first of all, I set up my cross. And so now I want to know my genotypic ratio. Well, to do that, I need to build my Punnett Square. Okay, so that'll be step 2. Okay, I'm going to do my Punnett Square. So I put my gametes go on the side, right? So let's say this is mom. She has the ability to pass on a big S and a little s. Dad has the ability to pass on a big S and a little s. So I put those on the edges. Okay, now I see what my actual offspring would be. So inside there, I have my offspring. And my question wanted to know my genotypic ratio. So remember, I just count, right? How many are homozygous dominant? Well, I've got 1. How many are heterozygous? How many are big S, little s? 2. How many are little s, little s? How many are homozygous recessive genotype? 1. So there's my answer right there. 1, 2, 1. Okay, let's look at another one. In humans, the ability to taste PTC is dominant to the allele for non-tasters. What is the percent? So I'm looking for a percent chance a child will be able to taste PTC. If the parents are a non-taster and someone that has a genotype big T, big T. Well, remember my first thing here is I need to set up my cross. Okay, so if I'm going to set up my cross, my parents are a non-taster. So non-tasters are recessive. So for my parent to be a non-taster, they have to be little t, little t. Otherwise, they will be a taster. If there's any big T's in there, they'd be a taster. So they're little t, little t. And the other parent is a big T, big T. Okay, so there's my other parent. Now I'm going to set up my cross, or my Punnett Square. I've set up my cross. Okay, so here's my square. I put my gametes on the side. This parent can only pass on a little t, little t. This parent can pass on a big T, big T. Well, the possibilities here for my children then are big T, little t, big T, little t, big T, little t, big T, little t. Well, my question wants to know my percent chance of a child that can taste it. And the ability to taste, my ability to taste is dominant. Well, every single one of these, that one can taste, that one can taste, that one can taste, that one can taste. 100% chance that there will be a taster. Because because it's dominant and all it needs is one dominant allele. Okay, so once I've done a monohybrid cross, I should hopefully be able to then move on to a dihybrid cross. And so a dihybrid cross is going to involve two traits. So now I'm not just talking about fur color. I'm talking about fur color and eye color. I'm talking about two traits happening at the same time. And so that lowers your probability that these things are going to happen. It's less likely for two things to happen at the same time as opposed to just one thing happening. So dihybrids are the same thing. You set them up the same as a monohybrid. You place your gametes, go on the edges. Okay, so my gametes would go here, my gametes would go here, and then my offspring will be in the middle. Okay, so I set it up the same way as a monohybrid. The thing you'll notice is that it's obviously a little bit bigger than a monohybrid. So let's take a look at how we're going to figure this out and how we're going to figure out the hardest part that people have with this is determining what the gametes are. So let's give an example here. Let's say I'm talking about these mice again, okay, and I've got gray fur. They're heterozygous for gray fur, and they're heterozygous for brown eyes. Brown eyes being dominant to green. So that's one parent. And the other parent, okay, my other parent is white and is also heterozygous for eyes. Okay, so this is my example here. And so the first thing I need to do when I find my gametes, I'm looking for all the possible combinations of alleles that they could pass on. Okay, so hopefully, I don't know if you guys have been doing this in math, okay, hopefully you're just doing distribution. And if you've used the FOIL method in math, this is very simple. Okay, if I group my like terms together, right, so I group my like terms together, my G's and my B's, I group them together, and so then I just distribute. I take my first term, so big G, big B. I take my outside terms, my outside, big G, little b. Then I take my inside terms, so little g, big B, and then I take my last terms, little g, little b. And now I have all my different allele combinations. Okay? I have all the different ways that this mom mouse, this girl mouse, can pass on her fur color and her eye color. She can either pass on a dominant for fur and a dominant for eyes, a dominant for fur and a recessive for eyes, a recessive for fur and a dominant for eyes, or she can pass on a recessive for fur and a recessive for eyes. So all of her different possible combinations. Okay? And then you go and you can do the exact same thing for the other mouse. Okay? I group my like, I've got to distribute my gametes. So I group my like terms. Okay? And then I just do my FOIL method. My first terms, so little g, big B. My outside terms, little g, little b. My inside terms, little g, big B. And then my last terms, little g, little b. So those are the possibilities that that male mouse can provide. And then we just put it together like we do, like you would for any other, for any monohybrid. Okay? Mom can pass on a big G, dad can pass on a little g, mom can pass on a big B, dad can pass on a big B. Okay? And I just work my way through the whole square doing this. Okay, I've got a big G that can be passed on, a little g, big B, little b. And I do want to group my alleles together based upon what they code for. So when I'm putting my genotypes together for my offspring, I do want to put my alleles for fur color together and my alleles for eye color together. I don't want those to be separated out. Okay, so I group them back together. Mom can donate a recessive, dad can donate a recessive, mom donates a recessive, dad donates a dominant. Okay, and like I said, we just go through and we fill in the entire square. And then once I've filled in the entire square, I can answer those prediction questions. Like, for instance, maybe I wanted to ask what percentage of the offspring would be heterozygous for both traits. So I just count. 1, 2, 3, 4. Okay, so I had a total of 16 squares. The answer to my question, heterozygous for both traits, is 4. So 1/4 of my offspring, or 25%, are going to be heterozygous for both traits. And so I answer it exactly the same as I would a monohybrid. I just have to remember, when I have 2 traits, I have to do this FOIL method. I have to distribute my gametes, have to do the FOIL method when I have 2 traits that I'm referring to. So with dihybrids, you're not going to have to do genotypic ratio for dihybrids, but you will have to do phenotypic ratio for dihybrids. So when you're doing phenotypic ratio for dihybrids, okay, you're looking for, remember, phenotype is what they look like. So when I'm doing phenotypic ratio here, I'm looking for my number that look dominant. So they look dominant for both traits. Okay, then I've got my number that look dominant, and then they look recessive. So they look dominant for the first trait, recessive for the second. Then my number that look recessive for the first trait, dominant for the second. And then last but not least, my number that look recessive for the first trait and recessive for the second trait. So let's look at an example of this. Okay, so let's say I've got, here's my cross down here. Okay, I'm heterozygous for both. And so I'm going to draw my square, draw my big 16-box square. And I've got 2 traits that I'm dealing with here. So I can't just split them up. To find my gametes, I have to FOIL, right? So I group my like terms, my first, big G, big B, my outside, big G, little g, my inside, oops, I wrote that on the wrong side, my inside, big G, big B, little g. Oh, I should have kept those. Don't pay attention to me right now. I should have kept those in the same order. Okay, little g, big B. And then my last, little g, little b. And I do the same over here. Okay, I'm going to separate, group together my like terms. And then I'm going to distribute. I'm going to use my FOIL method. Big G, big B. Okay, my outside, big G, little b. My inside, little g, big B. My last, little g, little b. And now I fill in my offspring. I take all of those, okay, and I bring them together. Big G, big G, big G, big B, big B, big G, big B, little b. And then I'm going to take this information that I have, and I'm going to look at their phenotypes. Again, so the phenotype is what will they look like. And they just need 1 dominant, remember, to look dominant. So I'm going to go through and I'm going to count how many look dominant for both traits. How many look recessive, or dominant for the first one and recessive for the second one. How many look dominant, recessive for the first one, dominant for the second one. How many look recessive for both. And that will be able to determine my genotypic ratio. Okay, so let's go through and let's count that real quick. So if I'm looking for how many are look dominant, 1, 2, 3, 4, 5. Oh, not that one. 5, 6, 7, 8, 9. So I have 9 that look dominant. Okay, it doesn't matter if they're homozygous or heterozygous, they still look dominant. Okay, so now I need to know how many look dominant recessive. 1, 2, 3, look dominant recessive. Okay, well, how many look recessive dominant? Okay, well, I've got 1, 2, 3,